photosynthesis. Essentially, it's converting some products. So it converts your CO2, right? Your carbon dioxide. It also converts the H2O into glucose, which is C6H12O6. As mentioned in the previous video about organelles, okay, and it also makes oxygen. Now, it does require sun in order for this reaction to occur. And photosynthesis doesn't just occur in plants. So it occurs in plants, obviously, but it also occurs in bacteria, as well as some protists and algae. Where does it take place? So it takes place in the chloroplast. Takes place in chloroplast. And within this chloroplast, it has a light absorbing pigment called chlorophyll. So I'll just write contains chlorophyll. Now, this example here that I just pointed out is pertaining to eukaryotic cells, not for prokaryotic cells. Are cells that have membrane-bound organelles. So things like protists, bacteria, algae, animals, plants, right? They all are considered eukaryotic. So anytime that we talk about chlorophyll being embedded within a chloroplast, we're talking about that. Now, in prokaryotic, though, organisms, they use free-floating chlorophyll, okay, in that cytoplasm. So this chlorophyll is not embedded within this organelle of a chloroplast. It's just free-floating in that jelly, that pudding, right? The cytoplasm that we talked about in the previous video of the organelle, where it keeps everything, all the other organelles, rigid and not moving around. Okay, and it's an area where a lot of chemical reactions occur. So that chlorophyll is free floating there, right? Chlorophyll free floats. So this photosynthesis formula that we just looked at, you can see that it looks very, very similar to the cellular respiration formula, but the cellular respiration formula goes in the opposite direction. So glucose being broken down by the oxygen that animals and even plants, right, and other organisms like protists, fungi, they use cellular respiration to produce carbon dioxide and water. But why? Well, those are the byproducts from what we really want to happen in cellular respiration. It's to make ATP. So I'll just draw this here on the side. ATP gets released, and this is the storage source for energy. When this P gets released and broken off from this adenosine triphosphate molecule, it releases energy, okay? And it also releases heat as well in the process. And it's actually because it releases so much energy and it's releasing oxides, right, like carbon dioxide, dihydrogen monoxide. These oxides are evidence that this is a combustion reaction, and obviously combustion releases a lot of energy and heat. This is happening in every single cell throughout your body, every single second, every single nanosecond. And we have enzymes to make sure that that cascading effect doesn't become very dangerous. And it ensures that the exergonic and the exothermic reactions are controlled in a step-by-step manner, so that it's not harmful within our body. So anyways, let's look at this in detail. So we have here a palisade leaf cell. So I'll just write maybe up here, palisade leaf cell. And on the inside of that leaf cell, let's focus in on the mesophyll, Why? Because this area is actually the primary site of photosynthesis. So maybe I'll write here as well. It's the primary, so one area, right, site of photosynthesis of, I'll just write PS, okay, of photosynthesis. Not photosystem, photosynthesis, the actual reaction. Now in every single one of these cells, right, so you see those cells here, right, there is, so let's focus in maybe on this one right over there, okay, that one, let's say that one is a chloroplast, okay, so each one of those mesophyll cells has an organelle of a chloroplast lying around. Very, very important. Why? Because within that chloroplast, guess what? If I were to take a zoomed in version, okay, of that uh, of that chloroplast, so I'm just gonna make it kind of look like that, okay? It has stacks of grana, okay? So kind of looks like that. And all of those put together is called a thylakoid, okay? So that is a thylakoid. And the areas around it, right there, right there, right there, okay, that space around it is called the stroma, okay, so this is called the stroma. And that's where a lot of the reactions are going to be occurring. There's actually two steps. So the first step, we have a light dependent reaction, okay, RxN for reaction. So this is the first phase. When the plant receives light, it uses that light in order to create two very important products. One of them is ATP, right, the storage site for energy, as well as NADPH. It's an electron carrier. It's a high energy carrier, very good at maintaining stability while it's holding on to the electron. And then from there, we need those products for the second reaction. Okay, the second reaction is the light independent reaction where it does not require light at this point. This typically happens at nighttime where light is not given. Well, I also forgot to say, when that light gets absorbed, right, within that thylakoid, right, inside the chloroplast, the ones that pick it up are certain proteins, as well as some pigments, and even some photosystems. We'll talk about that shortly. Anyway, so back to the light independent reaction. What is the point of it? Well, the point is to fix carbon. And I'll explain that in a diagram after I do a little brief summary for the first phase. Okay, so it fixes carbon and to make glucose. So that glucose will eventually be broken down with oxygen in order to carry out cellular respiration. Because plants also do cellular respiration as well. For cellular respiration. So I'll just put for cell resp, okay? Cell respiration. And this was light dependent, right? So light dependent reaction. We'll start from left to right like always. Maybe I'll put some reference over here, right? This is potential energy. Okay, so P stands for potential energy. It obviously increases going upwards. So when light comes in, as you can see over here, light's coming in here as well, right? Well, of course, when light comes in, that energy potential, that potential to be able to do work is obviously going to increase. So it's going to be increasing up that way, okay? And then we can have maybe a reaction progress going to the right. Okay, so I'll just put reaction progress. Okay, so like I said, sunlight gets absorbed by this little box over there. Okay, what's that called? We're going to call it a PS, all right? It's known as photosystem. Technically, it's actually called photosystem 2, okay, not photosystem 1. Photosystem 1 actually comes later over there, so maybe I'll write that there as well right now. That's photosystem 1. So photosystem 2 happens first. So it's a primary acceptor, okay? So it accepts it first. So once it receives that light, what is taking it? A specific area called the P680. So I'll put that over here, P680. Okay, it's a reaction center. So it takes that and releases the electrons. So I'll just put electrons over there, and the electrons eventually cascade to its next step. In order for this to happen, H2O, so H2O, let's just say it's floating around over here. This H2O needs to break up into its constituent parts. breaks up into H plus as well as O2 minus. And technically oxygen, you know, it's a diatomic molecule if you've taken chemistry classes already. Okay, so obviously it'll be an O2, not an O1. Okay, so because it's an O2, in order for it to balance out appropriately, if it's with the H, right, the H plus that's floating around, of course there needs to be two H's for every half of the diatomic molecule of oxygen, so that we're dealing with the law of conservation of mass properly. So if I had H2O over here, what we just did, right, the H2O breaks up into its constituent parts, right, balanced appropriately right now because we have one oxygen over there, and then we have two H's, over there. Here, I'll make it a little bit nicer. So our H2O breaking up, as you can see over there, into the H's and the O2s. Okay, so we got two of those. Anyway, so that's hydrolysis. Okay, so again, hydro, right, the water, lysis means break apart, so it's broken up into its parts. So now that electron can be moving down into its next step. Okay, it goes into the electron transport chain, right, the electrons are still there. To this over here, the electron transport chain. It's also coupled with a B6F complex to help bring that electron to its next step, right, the PS1. At this point, it's lost so much energy that it needs more sunlight, so more light, to help kick up that process, to increase the potential energy in order to do the work, so it can convert into its kinetic energy to continue to go along its subsequent steps. So the electrons get energized and are able to keep going. I want to note, at this point when that light got hit into this photosystem, a new reaction center called the P700 reaction center took up that energy and boosted that electron to continue going. Electron continues to go and eventually gets taken up by ferredoxin. Okay, I'm not going to write it here, but again, ferredoxins take the electrons with the help of an enzyme. I believe it's NADP reductase, which is absolutely crucial for the NAD pluses right now that are just floating around everywhere, to use those electrons and turn into NADPH. Like I said before, it is an electron carrier. Okay, so it holds that electron. Okay, all these electrons that have been getting to that step, it is now holding it. We need that for the next step when we get to the light independent reaction phase, right? The second phase of photosynthesis. So right now it's just holding them. So obviously the electrons right now, the count of electrons are decreasing. When the electron number decreases due to charges, the H pluses like this one, okay, they're going to be all around the place, right? H pluses, right? The hydrogen ions are going to build, okay? So obviously these are like protons, right? Because they're positively charged. So these H pluses, the protons, they continue to now increase. And where is it going to go? It now goes into the thylakoid space. On the inside here is the thylakoid space. So on the inner side of the thylakoid, these H pluses are going to go through due to that charge difference. Since these keep accumulating and we have no more electrons because these electrons got taken up by the NADPH. So the H plus are going to go through, okay? Within that thylakoid lumen, okay, right there. So it's going to go through, and this thing magic over here, this is known as ATP synthase, all right? ATP synthase. And it drives those H pluses through, and we have ADPs floating around, produces ATP and the H pluses. And again, all these reactions that are kind of going on, all of this is happening within the stroma, okay? All right, so the stroma's on the outsides over there, whereas, you know, the inner thylakoids over here are on the inside. So now we're in the stroma. Where are we going to use this guy and this guy? In the light independent phase. Let's get there now. So the light independent phase starts with a molecule here. It is 5 carbons with phosphates on either side. Okay, so the name of this thing is ribulose 1,5 biphosphate. In case you haven't taken organic chemistry yet, don't worry, this just means on the first and on the fifth, that's where the phosphates are located and held. Now there's 3 of these floating around, okay? Now, obviously we know plants, they like to breathe in CO2. So I'm going to go on to my next step where the CO2s now are going to be added on to this molecule. Okay, so I'm going to put this next phase is called the fixation. So this next step is called fixation, where carbon gets fixed, where 3 of the C's, the carbons, get added. So I'll just put here 3 C's added. Okay, so the CO2, maybe I'll bring it over here, the CO2, there's going to be 3 of them needed, right? Because we need 3 carbons. So we got 1 over there. We need to multiply by 3. So we have 3 CO2s coming in to add onto this ribulose 1,5-bisphosphate. Okay, 3 of those. And this is through the help of another enzyme called Rubisco. Again, we're not going to get into the specifics of like enzymes and all that because that's not the scope of this video. We're just looking at a general overview. So now let's take just one of these molecules, right? Instead of 5, we have 6, right? Because we got one of those carbons attached onto this thing, okay? Now, it's going to happen again. So another one of these is going to be right over here, and another one of these is going to be right below that, okay? But that one over here gets 1 carbon, and that one below that gets another carbon. So technically this is now times 3. So if you do all the math, it still all adds up. You have 1, 2, 3, 4, 5 carbons here, right? You times 3 gives you that 15. You add 3 of those, you get 18. Okay, so we got 1, 2, 3, 4, 5, 6 times 3 gives you 18 of the carbons. Same thing with the P's. Okay, the P's haven't changed at all. So we got 1P, 2P. So we got 2P times 3, you got 6 P's. Okay, 1, 2 times 3 still gives you that 6P. So at this point, we have 18 carbons. Now, if this continues to go as planned, we end up getting PGAs. Okay, these are acids, and this kind of gets split in half. Okay, so we get P, and then we have 3 of the carbons. All right, so we have 3 carbons there, and there's 3 of them, so times 3. And then these, right, are the other half, and we have 3 of them as well. So the math still checks out. They just got broken up into these, into an acidic formula, into an acidic molecule, rearranged. So it's still 18 carbons. So I'll just put over here, still 18 carbons. Now at this point, we start making something called G3Ps, okay? We start making 6 G3Ps. Technically, 5 G3Ps are actually made, but that 6th G3P actually becomes glucose and is used for cellular respiration. So that goes somewhere else. The 5 G3Ps that were made, in order for that to happen from the acids over here, is through the help of ATP, okay? Because it is a very energetic, it is a very energy taxing process for these acids to turn into the G3Ps. ATP gets broken up into ADPs, and it's actually 6 of those, okay? So it's a lot. Same thing with the NADPHs, okay? So we have NADPH, the electron carriers, breaks up into NADP plus. And again, 6 of those as well. Very, very taxing process. I'm just going to clean up this area to make it a little bit more neat. There we go. And then now we're going to bring those 5 G3Ps to start getting regenerated. Into what? Well, we want it to regenerate back into this molecule, right? So that eventually it's going to collect some even more CO2s, continue the process, make some more of that G3P to become that glucose, so that it can be sent off into the mitochondria or cytoplasm and help with cellular respiration. So I'll just put here to cell respiration. So how is it going to regenerate? Well, it needs to become this big molecule again. But this G3P here is obviously lower bonded, right? Compared to this big molecule, right? The ribulose 1,5-biphosphate. So it's going to release a lot of energy. Because when you need to form bonds, you need to release energy. So for this to form some bonds and become that big again, it needs to release energy. So this is an exergonic process. So again, ATP breaks up into its ADP, and this time there's 3 of them. Okay, very similar to what we did over here, that exergonic process, to turn into ribulose 1,5-biphosphate. Okay, I also forgot to mention over here we had 18 carbons, okay, up to that point, but remember we lost a few of them, right? We lost 3 of those carbons from that G3P. Okay, remember one of these became a G3P, and then the other 5 also became a G3P. It's just one of those G3Ps got sent off. So instead of having 18 carbons, we actually now only have 15. So I'll just put it in green here. So at this point we had 15 carbons, and this one that got sent off were 3 carbons. Alright, so we have 15 carbons over here. Even when it regenerated, it still became 15 carbons, right? Because we have 1, 2, 3, 4, 5 carbons times 3 still gives us that 15. So there's no problem with the math. And then this process just happens again and again and again. Now it could get a little bit more complicated if it didn't go through this process. Sometimes there are CAM plants or C4 plants that are not very effective, that do photorespiration, and so at this stage over here, instead of becoming those G3Ps, sorry, those PGAs, the phosphoglyceric acids, goes into another process which is way more inefficient. But for the realm of this course, just focus on this normal phase, quote unquote normal phase. So there we have it, that is photosynthesis. So I'm just gonna give a little bit of a summary of what we just learned. So we had the light reaction where the thylakoids took light to create the electrons. Okay, so it converted those photons, because that's what light is, right? Made them into the electrons so that it can go into the first phase that we looked at, the light dependent reaction. This happened within the stroma and then the inner thylakoid, the lumen. So the electrons converted the H2O into the 2 hydrogens and the oxygen. This is an endothermic reaction. It's obviously not exo, right? Because water doesn't have a lot of energy. It also has 0 calories, right? So it's not going to be breaking up and releasing much energy. Some of those H pluses converges with who? The NADP plus, remember? In order to output to make NADPH. The H pluses also help convert ADP in order to that ATP, which we needed in the light independent reaction like crazy. All while the oxygen gets released. Okay, and we'll need that oxygen a little bit later on. I'll explain why when we get to more in depth with the electron transport chain in cellular respiration next video. And then the light independent. Reaction uses that ATP and the NADPH with that carbon dioxides that get inhaled from the plant to make obviously glucose, the most efficient macronutrient.